Okay, so we are going to talk about nitrogen metabolism. So this will apply to both nucleotides and amino acids, because both nucleotides and amino acids have nitrogens in them. This lecture is mostly focused on how do we degrade amino acids and proteins, and we'll talk about nucleotides later. So the problem with metabolism and nitrogen is that we need to have some way to remove the nitrogen because it forms urea in our cells, and urea is toxic. So we'll talk about the urea cycle and how we remove the ammonia, the NH3, from the amino acids. We'll also talk about how we break apart proteins. There is a lot in this chapter, in this textbook, about specific enzymes, specific reactions, and we're not going to cover all of those specific reactions because there are 20 amino acids, which means there are 20 different pathways, many enzymes for both synthesizing those amino acids and breaking them apart. So you'll see a lot of examples. However, I am not going to ask you specifically how we deaminate glutamine or something like that. We're just going to kind of use a general approach because it could get very complicated. So the first thing that we're going to talk about is something called protein turnover. Protein turnover. So all proteins have a lifespan of maybe a few minutes or hours or days before that protein will naturally become degraded and then rebuilt. So this is called protein turnover. So we are continually replenishing our proteins. We're continually biosynthesizing and degrading them. So we need to get nitrogen from our diet, mostly from protein, because it's broken down and not replaced once these amino acids are turned over. So this protein turnover allows for this regulation. So your textbook says that the average half-life of a protein in a rat is 1 to 2 days. So take that for whatever it means. Some proteins last a few hours, some last a week or so. But on average, this is happening every couple of days. So the major way that proteins are degraded is through an enzyme. It's a protease. It's called the proteasome. So the proteasome, there's a picture in it on slide number 10 on the second of these two lectures. And again, I posted two nitrogen metabolism lectures just from two different textbooks. There's a lot of repeat, just a little bit of different information in each of these. So it's an ATP-dependent protease, and what it does is it breaks apart proteins. So proteins, when it's time for them to be degraded during this natural protein turnover, they will get targeted to the proteasome. And the way they get targeted to the proteasome is by a protein called ubiquitin. So ubiquitin is a protein. It's expressed in all cells. When it was first discovered, they discovered that ubiquitin was everywhere. It was ubiquitous, which was why it was named ubiquitin. A lot of times when proteins are discovered, they are named before we really understand everything that protein does. So ubiquitin was named ubiquitin because it was found everywhere. It was ubiquitous. The name ubiquitin doesn't have anything to do with its job, which is targeting proteins to the proteasome. So ubiquitin ligase is an enzyme, and it catalyzes the addition of ubiquitin to a previously conjugated ubiquitin. So what happens is you have a target protein. I'm just going to draw. Obviously, this is not what a real protein looks like. So you have the target protein. And on the target protein is a lysine. This ubiquitin is conjugated to the lysine. So this ubiquitin gets added to a target protein via a lysine on the target protein. Sometimes one ubiquitin is enough to send it to a proteasome. Some proteins are polyubiquitinated, meaning that many ubiquitins would be continued to be added here. Once these are added, then that tells this, it's a signal to send this whole complex to the proteasome. And again, there's a picture of the proteasome on slide number 10. And it has different subunits, and what it does is it's almost similar and opposite to the GROES chaperone system that we looked at. A chaperone is kind of like a barrel structure where an unfolded protein goes in and then the correctly formed protein comes out. The proteasome is kind of a barrel-shaped protein complex where a targeted protein, so this target protein, this has been targeted for, what's the word I want here? It's targeted for, to be broken apart, but that's not the word I want to use. Degradation, targeted for degradation. So the proteasome is where these proteins go that have been targeted for degradation. And what happens is those peptide bonds get broken inside the proteasome and individual amino acids are released. And then these individual amino acids are going to go, they'll stay in the cell, in the cytoplasm, but then at the ribosome, that is the site of protein synthesis. So we're constantly breaking apart proteins and making more. The site of protein degradation for these ubiquitin tagged proteins, these proteins that are ready to be degraded, is the proteasome. And then protein synthesis is going to happen on the ribosome. And this is coming later. We'll talk about protein synthesis, which is a process called translation. And that's coming at the end of the quarter. So that's the first major process that we'll talk about when we talk about protein turnover. Another thing that I want to talk about in this chapter is something called the urea cycle. So excess ammonia is transported to the liver for excretion as urea. So we need to remove ammonia. It goes to the liver to be excreted as urea in the kidneys. So the urea cycle is getting rid of that amino group, that ammonia group that we cannot metabolize. So this is called the Krebs Henseleit urea cycle. It's usually just called the urea cycle. It's on slide number 24 of the second of these nitrogen metabolism lectures. It shows an example of an amino acid, an amino group. So it actually starts with an amino acid, glutamate, and then that glutamate gets converted to something called N-acetyl glutamate. That N-acetyl glutamate then gets a carbamoyl phosphate. So carbamoyl phosphate is important in the ornithine transferase that's happening. So we're in the mitochondria is where this process is happening. It's happening in the mitochondria, between the mitochondria and the cytosol. So what happens is we go through successive reactions to remove the nitrogen group. We remove the whole nitrogen containing functional groups and produce urea. So that's the ultimate goal here, is to produce urea from removed amino groups. And again, this is on slide number 41. So this is important because without this to remove urea, urea toxicity would build up. So it's important to remove the toxic nitrogen containing parts so that we can then recycle the carbon backbones and use them for other things. So the next slide talks about the degradative fates of the amino acid skeletons. So now this comes back to the regulation and integration of metabolisms. So if you look at the TCA cycle, this is on slide number 25. So this is the fates of amino acid carbon skeletons. So we've removed the amino groups with the urea cycle. We're left with these carbon skeletons. Now these carbon skeletons can be intermediates in the TCA cycle, or they can be fed in at different locations in the TCA cycle. So there's examples, there's pictures. This is on slide number 25, and they're what are called glucogenic amino acids and ketogenic amino acids. So the glucogenic amino acids are amino acids that could then go through gluconeogenesis to produce glucose, and the blue amino acids, the ketogenic amino acids, can go through different pathways to become ketone bodies. And then also on that slide, there's some purple that can undergo both. So you are not responsible for which amino acid falls into which category, just that the carbon skeletons of the amino acids are then used as intermediates in the TCA cycle. Again, because there are 20 different amino acids, there are so many different situations happening here. Your text does go through some specific examples. One thing that I want you to know is that there is a difference in the amino acids. There's two classes of amino acids. There's called essential amino acids and non-essential amino acids. So the 20 amino acids fall into these two categories. Essential does not mean that they are important, and non-essential means they're not important. Essential means we cannot synthesize this group of amino acids, so it is necessary that we get these amino acids from our diet. Non-essential amino acids does not mean that these amino acids are not important. It just means that we can synthesize these amino acids. So it's not essential that we get them from our diet. They still are required for the proteins that we need. And these non-essential amino acids can be synthesized from intermediates in glycolysis and the citric acid cycle and all of those. And those can be found on the slides too. Again, I'm not going to ask you specifics about each of these pathways because each of these pathways has 2 or 3 or 4 steps, and there's 20 amino acids. So it just becomes too much material to cover in a course like this. But this is what I want you to know about nitrogen metabolism. I want you to know that protein turnover is facilitated by ubiquitin in the proteasome. I want you to know that it's necessary to remove the amino group from the amino acids because urea is toxic. We excrete it through urine, through our kidneys, and the carbon skeletons of those amino acids can then be fed in as intermediates on the TCA cycle, and essential amino acids cannot be synthesized, so we have to get them from our diet. Non-essential amino acids are just as important as the essential amino acids. We just have enzymes that we can biosynthesize them ourselves.